Hello there, and welcome to the Whispering Science, your gentle guide through the peaceful landscapes of science. Now, I do hope this isn't too interesting that it keeps you awake. But if it does, well, at least you'll have learned something fascinating about how DNA writes your story, how the tiniest threads within your body carry the record of what makes you, you. Before we begin, if you enjoy these slow science journeys, I'd be ever so grateful if you tapped the like button, subscribed, or even just left a comment telling me where in the world you're watching from and what time it is in your corner of the night. So, settle in, get comfortable, and let's begin our quiet voyage into the mysterious, beautifully ordered world of DNA, the molecule that not only shapes your existence, but quietly hums its tune every moment of your life. As you breathe in and exhale out, picture for a moment the entirety of your body, head to toe, finger to finger, skin, muscle, and bone. Now imagine shrinking down, not just a little, but until you're smaller than the smallest cell. At this scale, the world is very different. There are no organs or tissues, only a soft, gentle expanse filled with countless living cells, each invisible to the naked eye, and each humming with life. Every single one of these cells, so numerous that there are about 37 trillion in your body, holds a tiny library, a set of instructions so compact and complete that if unraveled, it could stretch to the moon and back many times. Tonight, we're going on a gentle exploration inside that microscopic library, discovering the secrets within your DNA. We'll learn the story of its discovery, the remarkable complexity it holds, its role as the silent conductor of your biological orchestra, and how it connects you to the patterns of your family, your ancestors, and even to every other living thing on Earth. As we move softly through this tale, you may begin to understand just how remarkable it is that all that you are, from the shape of your nose to the twinkle in your eye, is quietly written and rewritten in a code carried within each cell. But most importantly, I invite you to relax, to let go of the day's cares, and allow curiosity, no matter how gently it flickers, to carry you along. So here we drift into the secrets of your DNA, together on this peaceful night. Let's float deeper now into the wondrous blueprint inside you. If you've ever assembled a piece of furniture or followed a recipe, you'll know that instructions are everything. DNA, short for deoxyribonucleic acid, is your body's ultimate instruction manual. Unlike an ordinary booklet you might keep in a kitchen drawer, this manual is written in a special chemical language and carefully tucked into the heart of each of your cells. The instructions it contains determine whether you'll have blue eyes or brown, curly hair or straight, and even details as intricate as your body's resistance to certain illnesses or the rhythm of your heartbeat while you sleep. Each instruction in this blueprint is carried out with astonishing precision and complexity, all to build and maintain the endlessly intricate organism that is you. Now, imagine building a model airplane. The box contains hundreds, even thousands of tiny pieces, yet a simple, clear plan guides their arrangement, so the finished plane takes to the air rather than falling apart. Your DNA does something even more dramatic, beginning before you ever took your first breath and continuing every day across your entire lifespan. The blueprint in your DNA is written in a code billions of letters long, and yet, amazingly, this entire instruction set fits within the nucleus of each microscopic cell, a space less than a few millionths of a meter wide. Your DNA's message isn't just about what you look like, although that's where much of its magic is visible. It also describes in fine detail how your heart should pump, how your bones should grow, and how your immune system is to recognize invaders and keep you safe. It's responsible for whether you can roll your tongue or taste certain flavors, how tall you might become, and even how you respond to caffeine. Scientists have calculated that if you typed out the entire DNA code for the average human, it would fill around 262,000 pages of text. That's more than 400 standard-sized books. And yet, nature packages all of that within you with a grace that feels almost magical. This blueprint has another clever feature. While the core design is unmistakably human, the instructions vary subtly from person to person. It's these variations, tiny differences in how certain parts of the code are written, that give rise to our endless diversity. From your skin's ability to tan, to your tendency to laugh loudly or quietly, little spelling differences in your DNA help shape your temperament, your tastes, and how you interact with the world around you. But your DNA blueprint isn't a static, dusty book. It's more like a living document, subject to small edits and tweaks as life goes on. Every time a cell divides to create new skin, hair, or blood, the DNA within must be copied faithfully. Sometimes, tiny typos happen. Some make no difference at all, while others might bestow new abilities, lead to inherited conditions, or simply form the vast and beautiful tapestry of human difference. It may be tempting to imagine your DNA as a long, unspooling ribbon, but the reality is even more efficient. Folded, compacted, and masterfully organized, your DNA sits as tightly coiled strands within the cell. If you could carefully extract and line up the DNA from just one of your cells, it would stretch about 2 meters, over 6 feet, from end to end. Now multiply that distance by the estimated 37 trillion cells in your body, and you'll find there are billions and billions of kilometers of DNA within you. More than enough to wind around the Earth's equator thousands of times. Astonishing, isn't it? Such a grand blueprint, written out billions of places and yet unique in its own little variations each time. As we settle more deeply into our exploration, remember, every part of you, from the cells in your skin to the deepest, most protected bones, carries a set of instructions as complete and intricate as a symphony. Sometimes the music is familiar, a tune played for millennia in the animal kingdom. Other times it's your own special melody, a unique story composed just for you. So, as you ready yourself for the next part of our tranquil scientific journey, know that the story written within your cells is both unimaginably vast and intimately yours, a testament to the delicate, unfathomable complexity that makes each single life utterly irreplaceable. Let's drift backwards now, gliding gently through the currents of time to witness a pivotal moment in the history of science, the discovery of DNA's beautiful, spiraling form. Before the mid-20th century, scientists suspected that our genetic information must be stored somewhere inside our cells, but they could not yet glimpse the tiny molecules themselves. It was as though humanity had a locked book containing its most precious secrets, with no key to open its cover. The search for DNA structure was filled with both curiosity and quiet competition, with several brilliant minds working late into the night, their hypotheses sometimes as tangled as the mysterious substance they sought to understand. It was in 1953, in the bustling, rain-soaked city of Cambridge, England, that two scientists, James Watson, a young American biologist, and Francis Crick, a British physicist, finally cracked the code. Their inspiration arrived, in part, thanks to crucial X-ray photographs taken by Rosalind Franklin, a meticulous English chemist, whose data provided key insights into DNA's physical form. In her photograph, often called Photo 51, X-rays diffracted through the DNA molecule and created a cross-shaped pattern, hinting at helices wound together. This image is sometimes considered one of the most important photographs in the history of science, even though Franklin herself remained under-celebrated during her lifetime. Watson, Crick, and their colleagues sketched and modelled, twisted and retwisted wire frameworks, and after months of persistent inquiry, they imagined the shape that would change biology forever, the double helix. Picture a twisting ladder with sides formed by sugar and phosphate backbones, and rungs made of paired chemical bases. Each twist of the ladder is graceful and regular, repeating every 10 rungs or so. The structure suggested not only how DNA might carry information, but also how it could be unwound and copied whenever a new cell was born. Their discovery, published in the scientific journal Nature in April 1953, rocked the scientific world. It was an orchid in a field of wildflowers, a moment of pure insight that changed our understanding of life. This elegant shape wasn't just a lucky guess. It satisfied the rules of chemistry and geometry and, crucially, explained how genetic information could be stored and transmitted with such astonishing fidelity. Because the two strands in the helix are complementary, each base on one strand is always paired with its specific partner, genetic instructions could be duplicated almost perfectly whenever a cell divided. Like a zipper opening and closing, the helix could gently unwind, letting each side guide the recreation of its twin. This became the central model for how heredity works. DNA provides a code, exactly duplicated and easily shared from one generation to the next. The Nobel Prize Committee took notice, and in 1962, Watson, Crick, and Maurice Wilkins, another crucial researcher from Franklin's laboratory, received science's highest honor. Tragically, Rosalind Franklin had died of cancer just 4 years earlier at the age of 37, having never been formally recognized for her foundational contributions. Over time, however, her role has become a central and celebrated part of this great scientific story. Why does the shape matter? Imagine a ladder that can coil up so tightly it fits in a shoebox, but can also unroll instantly to span the length of a room. DNA's double helix gives it both strength and flexibility, allowing it to be packaged tightly inside chromosomes, yet remain accessible for reading and copying. In fact, this spiraling, repeating structure is preserved with only small variations across almost every living thing, from the tallest redwoods to the tiniest bacterium to every animal, including you. Today, over 70 years after its revelation, the image of the double helix is almost iconic, a swirl that you might see decorating research labs, classroom posters, and even jewelry. But the beauty runs deeper than any picture. It is in the quiet mathematical order of nature, the way simple chemical units repeat, spiral, and combine in endless stories. The double helix is not just the mark of human life, it's the signature of biology's unity, of common descent and shared origins from creatures that lived long before us. As you relax deeper into your evening, think of how the discovery of this gentle, twisting staircase in your cells forever changed what it means to be human. With every heartbeat, every moment of growth, and every inherited trait, the double helix serves as a reminder of life's quiet ingenuity, a testament to discovery, teamwork, and the patient unraveling of the universe's most persistent mysteries. Now, let's soften our focus and gently turn towards the tiniest alphabet in nature, the 4-letter genetic code that spells out the story of you, one word and sentence at a time. If you've ever sent a message using only a handful of letters, maybe one of those word games, or even just texting with only emojis, you'll know that a small alphabet can build a vast vocabulary. Inside your DNA, this idea is taken to its beautiful extreme. The entire universe of human biology is composed from just 4 simple chemical letters. These letters aren't shaped like the A, T, C, and G that we write on paper, but biochemists use those labels as shorthand for the 4 chemical building blocks of DNA. The full names are a bit of a mouthful: adenine A, thymine T, cytosine C, and guanine G. Each is a special kind of molecule called a nucleotide, and together they form the rungs of DNA's famous twisted ladder. Each position along the DNA chain holds just one of these 4, but when arranged in different orders and lengths, they can give rise to every living detail of every living thing: your laughter, your height, your ability to digest certain foods, and so much more. It's remarkable to think that the recipe for an entire person can be written using just these 4 chemical characters. In the human genome, these bases are arranged in pairs. A always pairs with T, and C always pairs with G, like partners in a delicate molecular dance. This strict pairing, one A always matched by one T and one C with one G, ensures that the genetic message stays stable and readable across generations. You might wonder how such a tiny alphabet could build something as complex as a human being. The key lies in combinations. Just as the English alphabet can make immense stories and uncountable sentences from only 26 letters, so too can your DNA spell out the instructions for making eyes, skin, muscles, and memories from these 4 bases. Strings of bases combine into words that scientists call codons, set sequences of 3 bases like ATG or CGA. Each codon acts like a tiny instruction, calling for a specific building block of life, an amino acid, the fundamental unit of proteins. And just as punctuation marks can steer the meaning of a sentence, in DNA there are special codes to signal "start here" and stop here, making sure the story unfolds in the right order. The order, or sequence, of these 4 bases in your DNA strand is what defines everything, turning the simplest code into the most elaborate architecture in nature. For example, the gene that controls your blood type might be a few thousand bases long. The gene for insulin, so critical in your body for controlling blood sugar, is only about 1,400 bases, yet its precision is essential for your health. Little by little, scientists have learned to read this molecular script. Think of sequencing machines, fast, tireless readers, scanning the 4-letter sequence, uncovering personal stories written across billions of letters, as effortlessly as reading sheet music for a symphony. Since the turn of the millennium, the ability to read this code has gone from slow and costly, weeks or years to decode even short fragments, to today's reality, where an entire human genome can be sequenced in just a day or two, with costs dropping every year. Perhaps, as you rest, you'll marvel at how different you are from those around you, yet how astonishingly similar too. For every 1,000 base pairs, on average, only 1 varies between any 2 people on Earth. That's less than 0.1% difference, yet it is enough to account for your unique smile, the way you see colors, or how your body responds to a virus. And it's this same 4-letter alphabet, shuffled, repeated, and gently altered, that also builds the tall trees, the mewing cats, and the fruit on your breakfast table. In many ways, the alphabet of life is a lesson in unity amidst diversity and complexity from simplicity. Its message, repeated again and again all across the natural world, is that life itself is built on subtle variations in a shared song. So as we journey forward, keep in mind how these humble 4 chemical letters quietly weave together the story, not only of who you are, but of all living things, an invisible thread connecting you to the very roots of life on Earth. Billions of bases, 1 you. Let's pause here and contemplate the incredible scale on which your genetic story is written. Sometimes, when we hold a novel in our hands or gaze at a painting, we're struck by the sheer number of tiny, deliberate details that combine to make a unified whole. The same is true of your DNA, but at a scale and complexity that easily surpasses anything crafted by human hands. To appreciate just how extensive your genetic code is, imagine each of those 4 chemical letters, A, T, C, and G, lined up in a dizzyingly long chain. In total, the human genome contains roughly 3 billion pairings, or base pairs, organized in a precise and unique sequence for nearly every person on Earth. It's almost impossible to truly grasp such a large number. If you were to try reading your genome aloud, pronouncing just 1 letter per second, non-stop without sleeping, it would take you over 95 years to finish the task. Written in printed books, your genome would fill shelf after shelf in a quiet library, a testament not to fiction or poetry, but to the script that makes you possible. And astonishingly, this entire vast archive fits into the core of a single-cell nucleus, so exquisitely coiled and packaged that it's invisible, even with the most powerful light microscopes. The scale becomes even more awe-inspiring when you remember that nearly every cell in your body contains its own full copy of this 3 billion letter story. Think about your skin. The cells there are constantly dividing, sloughing off, and being replaced. Each new cell needs a perfect copy of this code, so it can do its job to protect, to heal, or to differentiate into hair, sweat gland, or nail. Multiply those 3 billion base pairs by the estimated 37 trillion cells in your body, and the total number of DNA bases inside you is unfathomable, somewhere in the realm of 111 quintillion, or 1.1 x 10 circumflex 20, if you prefer scientific notation. It's a number vaster than all the grains of sand on all the world's beaches. This grand scale is more than a mere curiosity. It's a reflection of the complexity required to build a living being. Across these billions of bases, instructions must be placed, not too close or too far apart, for every structure and function. Most of these codes reside within genes, but much of the genome is made up of stretches that, for a long time, scientists called junk DNA, because they didn't code for proteins. That term has begun to soften. We now know that many of these stretches have subtle regulatory functions, acting as switches to turn genes on or off, or contributing to how different cell types appear and behave. But vastness does not mean sameness. While 99.9% of your DNA is identical to everyone else's, the 0.1% that is different across individuals provides nearly infinite possibilities for variation. Each base that is switched, added, or deleted, sometimes in cascades of hundreds or thousands, can shape familiar traits like your eye color or subtle things like your sense of smell. These changes, called variants or single nucleotide polymorphisms, mean that even among billions of people on Earth, your DNA is essentially one of a kind. It's your own private code, foundational, yet ever so slightly distinct from all others. The sheer volume of information contained in your DNA also means there's room for resilience. Let's imagine a typo in an instruction manual. With so much information, occasional errors don't always end in disaster. Many changes or mutations don't affect you at all, either because they're in a region that isn't read often, or because the code is surprisingly robust, with overlapping instructions and a bit of built-in redundancy. There are, after all, 64 possible codons, but only 20 amino acids to make up proteins. This means your genetic script can weather minor changes, allowing evolution to experiment, adapt, and refine over generations without putting you at constant risk. From birth to old age, your cells make countless copies of this 3 billion letter script, sometimes flawlessly, sometimes with tiny shifts that echo quietly through the generations. The vastness of your DNA is a tribute to the complexity of life, but also a reminder that deep within you, order emerges from unimaginable numbers. It's humbling, soothing almost, to realize that you are the product of billions upon billions of molecular decisions, each one a letter in the vast silent story that is only yours. So as you rest, remember, you carry a vast library in every cell, a long, detailed, and beautiful narrative written with 4 letters across billions of pages to create a single, irreplaceable you. Let's now ease into the heart of your genetic narrative and discover how genes, those specialized segments of DNA, truly make you, you. If DNA is the great instruction manual, then genes are the individual recipes, each detailing how to create a unique ingredient or component for the masterpiece that is your body. Imagine strolling through an old library, shelves upon shelves of books lining the walls. In this quiet collection, every volume represents a distinct gene, and each gene contains the detailed directions for assembling a particular protein. Molecules that execute nearly every function required for life. In more technical terms, a gene is a stretch of DNA where the sequence of A, T, C, and G letters carries the code for making a particular protein or sometimes a functional RNA molecule. Think of proteins as the workhorses of your cells. They construct your muscles, give structure to your hair and nails, digest food, carry oxygen, trigger immune responses, and much more. Each protein is built from chains of amino acids, and the way those amino acids link up and fold determines its purpose and shape. It's your genes that specify exactly which amino acids are needed and in what order, a unique recipe for each critical function. Humans are estimated to have around 20,000 to 22,000 genes, scattered across their 23 pairs of chromosomes. This sounds like a lot, and it is, but remarkably it's fewer than in some seemingly simpler organisms, such as the humble water flea or certain plants. So, while numbers matter, the complexity of human beings comes from how our genes interact, how they are regulated, and the astonishing diversity of proteins that can be created from a single gene through a process called alternative splicing. In alternative splicing, the same gene can generate multiple different proteins depending on how its instructions are read and processed, a bit like reading the same recipe with different options for each meal. Your genes are both architects and organizers, giving rise not only to obvious outward traits like height, hair color, or dimples, but also to hidden processes. Take, for instance, the gene that enables your hemoglobin to hold oxygen in your red blood cells, or the gene that instructs your pancreas to make insulin for managing sugar in your bloodstream. Some genes, known as regulatory genes, oversee the activity of others, serving as conductors to synchronize the orchestra of cellular activity, making sure that everything happens in just the right amount and at the right time, during childgrowth, healing after injury, or responding to stress. This exquisite organization helps explain why, though all of your cells contain the same DNA, only certain genes are switched on in each cell type. For example, the genes responsible for producing digestive enzymes are active in your stomach, while those needed to contract muscle fibers are only expressed in your muscles. It's an elegant system, one that ensures each part of your body receives just the instructions it needs, no more and no less, for the job at hand. But what happens if a gene contains a variation, a difference in that long chain of DNA letters? Sometimes these differences are neutral. Sometimes they give rise to new traits or abilities, and sometimes they may lead to inherited conditions. A single letter change in the gene responsible for hemoglobin, for instance, can cause sickle cell anemia, while slight variations in other genes can influence characteristics as subtle as your caffeine metabolism or the way you perceive bitter flavors. There are even silent mutations, variations that seem to have no immediate effect at all, but can still be passed down and occasionally reveal their impact generations later. Thanks to advances in genetics, scientists can now identify which genes are tied to certain conditions or traits, unlocking secrets that were unknown even a few decades ago. Whether it's the color of your eyes, your predisposition to certain medical conditions, or your natural talents and quirks, these are all shaped by the gentle, ongoing work of your genes. They are tiny custodians, silent, precise, and incredibly wise, crafting your unique story every single moment. So as you slip deeper into the tranquility of the night, reflect on this. Within you are thousands upon thousands of genes, quietly crafting the mosaic of your body, your health, your quirks, and your very being. Each tiny segment, every letter, contributes to the rich and irreplaceable composition that is you. Let your thoughts meander now to the extraordinary way your DNA is stored, neatly and elegantly packed into structures called chromosomes. Just as a precious heirloom or an important letter requires careful storage to remain safe across the years, your incredibly long DNA strands must be managed to fit perfectly within the confines of your cells. Visualize, if you will, an impossibly long ribbon, over 2 meters, more than 6 feet from each cell, somehow tucked with the utmost order into a nucleus barely a few millionths of a meter wide. This everyday miracle is accomplished not by magic, but by remarkable biological engineering, with chromosomes as the chief organizers. A chromosome is essentially a single, continuous DNA molecule, coiled and looped many times around specialized proteins known as histones. These histones act like tiny spools, winding up the DNA thread until it is compact, yet always able to be unwound when the genetic code needs to be read or copied. This packaging allows for efficiency and accessibility, much like storing a massive novel in a compact, accessible format, ready to be opened to any page at a moment's notice. Human cells typically contain 23 pairs of chromosomes, for a total of 46 in almost every cell. You received one set of 23 from your mother and one from your father, which means your chromosomes are half maternal, half paternal, a harmonious duet of ancestry. These chromosomes are numbered from 1 to 22, which are called autosomes, and then the 23rd pair consists of the sex chromosomes, labeled X and Y. The combination of these sex chromosomes determines your biological sex, XX for female, XY for male. Yet, as science gently reveals, even these labels represent a spectrum of biology with exceptions and surprises. The chromosomes aren't all the same size. Chromosome 1 is the largest, holding about 249 million base pairs, while chromosome 21 is one of the smallest, with just about 48 million base pairs. These size differences are significant. Larger chromosomes typically carry more genes, and variations in these chromosomes can be linked to genetic conditions. For example, Down syndrome, first clearly described by physician John Langdon Down in 1866 but only genetically explained in 1959, results from having a third copy of chromosome 21, a tiny difference in packaging that changes development in profound ways. During most of your life, chromosomes aren't neatly visible under a microscope, but exist as loose fibers in the nucleus, a relaxed state called chromatin. However, as a cell prepares to divide, this chromatin condenses, forming the classic X-shaped structures you might recall from science textbooks. Each X-shape reflects two identical DNA copies joined together, which will soon be separated into new cells, ensuring that every cell receives a complete set of instructions. The way DNA is folded within chromosomes does much more than save space. It also controls which genes are active at any given moment. Specific regions of chromosomes can be more tightly packed, making genes in those areas hard to access, while other regions are more open, allowing the cell's machinery to read the genes and produce the right proteins for that cell type. This organization is so precise that two different cell types, say a nerve cell and a skin cell, can access entirely different sections of identical DNA, simply due to how the DNA is folded and which chromosomal areas are open or closed to the cell's protein-making machinery. Even the ends of chromosomes hold special genetic tricks. These ends are called telomeres, acting rather like the plastic tips on shoelaces, protecting the chromosomes from fraying or sticking to each other. With each cell division, the telomeres shorten a little, serving as a biological clock of sorts. Scientists like Elizabeth Blackburn, who won a Nobel Prize in 2009, have studied telomeres and their link to aging and diseases. Our understanding of chromosomes continues to deepen, and we now see them not just as passive storage units, but as dynamic, responsive organizers shaping how genes are used across time and in response to life's changing demands. So, as you drift gently onward, imagine all the chromosomes in your cells, each a marvel of folding, organizing, and preserving your unique genetic code. They are the silent librarians and curators of your personal archive, keeping your story intact as you grow, heal, and live. Allow your thoughts to drift softly along the threads of time as you consider the family legacy interwoven within your DNA. There is a profound comfort in knowing that much of what makes you unique is not randomly assigned, but gently handed down, generation after generation, by those who came before you. Your DNA carries echoes of your parents, your grandparents, and your distant ancestors, each contributing a crucial part to your story. This inheritance, called heredity, is the delicate process by which genetic information is passed from one generation to the next, forming a living bridge between the past, present, and future. To understand this delicate orchestration, imagine your mother and father each bringing half of their own vast library of DNA to create you. During the formation of egg and sperm cells, a process known as meiosis, the 23 pairs of chromosomes in each parent are separated, and one chromosome from each pair is randomly selected to be passed on. This selection mixes and matches parental DNA in billions of possible combinations, ensuring that even siblings, except for identical twins, are genetically unique. When the egg and sperm unite, they form a new combination, one complete set of 23 chromosomes from your mother and another set of 23 from your father, reuniting into 23 pairs and setting the foundation for a new, singular you. It's not just whole chromosomes that are shuffled. Within each chromosome, segments can be exchanged in a process called crossing over. Here, during meiosis, matching chromosomes from the mother and father physically intertwine and swap equivalent pieces in a choreography that introduces even more genetic variety. This recombination generates entirely new combinations of genes, blending not only your parents' traits, but bits and pieces of their own ancestors as well. This is why you might inherit your grandfather's chin, your grandmother's hair color, and your great-aunt's infectious laugh, sometimes even when those traits seem to skip a generation. Many traits are influenced by just a few genes, such as the color of your eyes, or whether your earlobes are attached or detached. But most qualities, your height, the tone of your voice, your tendency to freckle, or your risk for certain conditions, depend on the interplay of many genes, as well as subtle influences from your environment. These are called polygenic traits, and they are the result of hundreds or even thousands of genetic instructions working together, colored by the life experiences your ancestors and you have collected. Your DNA also stores a record of your ancestral roots far beyond your immediate family. Within the molecule are tiny sequences that have been inherited, often unchanged, over hundreds or thousands of years. These genetic markers, scattered throughout your genome, are the reason commercial ancestry tests can trace your lineage, sometimes mapping geographic origins or identifying surprising connections to populations on the other side of the world. Mitochondrial DNA, from the energy-producing structures in your cells, is passed down almost entirely from your mother, while the Y chromosome, present only in males, is passed directly from father to son, offering special insights into maternal and paternal lineages. Through careful study, geneticists have painted astonishing portraits of human migration, revealing how ancient peoples spread across continents and adapted to wildly different environments. For example, certain gene variants help people digest milk into adulthood, lactose tolerance, and are common in populations with pastoral ancestry, a living echo of long-ago shifts in diet and culture. As you reflect tonight, feel the gentle tug of history written into your very body. Every cell is a quiet time traveler, carrying forward traits that survived wars, migrations, famines, and celebrations. Whether your eyes mirror those of a distant ancestor, or your hands resemble a loved one's long gone, these echoes in your DNA are reminders that you are both wholly yourself and intimately bound to a greater human story, spun across countless generations. So as you slip further into relaxation, let yourself imagine the tender, unbroken chain of inheritance that runs through your cells, a living memory, always present, always quietly unfolding, night after night. Settle further into comfort as we imagine the remarkable process by which your genetic story is faithfully copied and preserved. An exquisite copying ritual known as DNA replication. Every second of every day, in every growing, healing, or renewing cell in your body, this process quietly unfolds, ensuring that each new cell receives an exact duplicate of your irreplaceable DNA code. The precision required is breathtaking. When a cell needs to divide, whether to replace damaged skin after you scrape your knee or to generate white blood cells to fight off an infection, it first must replicate all 3 billion base pairs of your genetic alphabet with a fidelity that allows only the rarest of errors. To visualize this marvel, imagine the double helix gently unwinding, the two intertwined strands slowly separating like a zipper unfastened in perfect sync. Specialized molecules called enzymes, the caretakers of this operation, play graceful and vital roles. The enzyme helicase first pries open the helix, unzipping your DNA's tightly coiled ladder inch by inch. Next, DNA polymerase, the master builder, arrives, reading each exposed base on the original strand and selecting the perfect complementary base from the pool floating nearby. Where there's an A on the original strand, DNA polymerase brings over a T to match. Where there's a C, it brings a G. With astonishing efficiency, this enzyme propels new strands alongside the originals, pairing one base at a time, building the duplicate ladder before your eyes. Now, since DNA is so long and tightly packed in each cell, this copying process doesn't start at one end and drift all the way to the other. Rather, it begins at thousands of locations along the DNA simultaneously, with teams of enzymes working in parallel, like skilled librarians photocopying thousands of pages at once. In human cells, the entire job of copying all 3 billion bases can be done in just a few hours, a pace that puts even the speediest assembly lines to shame. As each section is finished, the new DNA strands coil up and settle into the growing new cell, ready to pass on every detail of the vast genetic legacy. DNA replication is so accurate that on average, there's only about one copying error in every 10 billion bases added. This is thanks not only to the precision of DNA polymerase, but also to a suite of proofreading enzymes that review the new strands, scanning for any misplaced letters, and making instant repairs. Without this molecular oversight, the entire system would break down, and life's continuity would falter. Yet despite such careful editing, tiny mistakes, known as mutations, occasionally slip through. Sometimes a single base is copied incorrectly, or a letter may be skipped or duplicated. Most of these changes are harmless, as much of your DNA either has built-in redundancy or resides in non-coding, less critical regions. Occasionally, a mutation may cause a new trait to appear, or more rarely, may lead to diseases like certain cancers that arise when the instruction manual becomes jumbled in crucial places. It is these rare changes, enduring over generations, that not only fuel the slow march of evolution, but also contribute quietly to the rich diversity you see in all of life around you. The crystal clarity and reliability of DNA replication form the bedrock of all tissue growth, healing, and regeneration. Each time your body knits a wound, grows a hair, or recycles a cell, this ancient copying process is at work. Alongside this diligence, your cells are always adapting, sometimes incorporating rare errors that may one day provide an advantage, or simply serving as reminders of the system's perfect imperfection. As you linger here in thought, know that billions of cells within you are remembering, copying, and gently renewing your genetic blueprint every moment of your life, even as you drift peacefully in bed tonight. Their silent, seamless work writes the next pages of your story, ensuring the harmony and continuity of who you are, cell by cell, night after night. As you relax more deeply, let's explore the journey from your DNA's silent script to the bustling world of proteins and action, what biologists call the central dogma, the very heart of genetic expression. Imagine now that your DNA is filled with instructions, but those instructions remain quietly tucked away inside the cell's nucleus, like a cherished cookbook sealed behind glass. For your body to benefit from its wisdom, these instructions must be read and put into practice. This process unfolds in a two-step dance. First, your DNA's language is carefully transcribed into a temporary readable message, and then that message is translated into proteins, the vital actors that shape every structure and activity within you. Let's gently follow this journey, beginning with transcription. Inside the nucleus, specialized enzymes locate a gene, just a small segment of the entire DNA strand, and begin to read it. To do this safely, without disturbing the precious master blueprint, your body makes a disposable copy in the form of a molecule called messenger RNA or mRNA. This mRNA carries the same genetic message as the DNA, but with one important twist. Instead of thymine, T, RNA uses a slightly different base, uracil, U. This subtle shift ensures the copy is recognized as temporary, meant to be used and then replaced, so your original DNA remains guarded and unchanged. Once this message is complete, the mRNA gracefully exits the nucleus and enters the cell's cytoplasm, the busy interior where proteins are assembled. Like a well-marked delivery, this strip of genetic code finds its way to a ribosome, which you can imagine as a tiny, precise molecular chef. This ribosome now begins the translation phase, reading the mRNA's genetic recipe 3 letters or bases at a time. Each trio, remember, is a codon, specifying a single amino acid. There are just 20 basic amino acids, yet the many combinations and sequences the ribosome reads allow for tens of thousands of different proteins to be built, each with a unique shape and function. This act of translation is astonishingly fast and accurate. As the ribosome slides along the mRNA, it pieces together amino acids in the precise sequence specified by the gene, like threading beads onto a string, one after another. As the amino acid chain lengthens, it begins to fold, spontaneously and elegantly, into a three-dimensional structure. The result, a unique protein, perfectly suited to its task, whether forming the keratin in your hair, the collagen in your skin, or the enzymes driving every chemical reaction in your cells. Each tiny step in this process is crucial for life. If a single letter is off in the original DNA, or the transcript is mismatched, the finished protein might be slightly altered or completely non-functional. Errors can be harmless, or occasionally, they may lead to one of thousands of known genetic disorders. Yet for the vast majority of your body's tasks, growth, healing, metabolism, defense, the system works with barely a hitch, thanks to hundreds of millions of years of evolutionary refinement. Historically, the realization of this central dogma was a breakthrough that transformed biology. Francis Crick, after helping uncover the double helix, proposed this flow of information, from DNA to RNA to protein, an idea proven and elaborated through decades of meticulous research. In the latter half of the 20th century, experiments by scientists such as Marshall Nierenberg and Gobind Khorana cracked the genetic code, revealing exactly which codon meant which amino acid, a puzzle nearly as elegant as DNA's structure itself. But the wonder doesn't end with the simple creation of proteins. Your body manages thousands of simultaneous transcription and translation events at every moment, all perfectly timed and coordinated according to your needs, environment, and stage of life. Certain genes are only expressed during childhood, others mainly when you're ill or under stress. The central dogma, though unwavering in its main flow, has 1,000 subtle regulatory switches and variations, ensuring that each cell produces the proteins it needs, no more and no less. As you nestle deeper into quiet, let yourself marvel at this inner ballet, your DNA setting forth gentle messages, your mRNA faithfully carrying them forward, and your ribosomes building the molecular machinery of life, over and over, minute by minute, night after night. All this hidden from your everyday awareness, working ceaselessly to keep you alive, adapting and maintaining your being as you rest in peace. Let's relax further as we turn to the quiet drama of mutations, the tiny, often invisible changes in your DNA that can have surprisingly large impacts. Even as DNA replication masterfully copies your genetic code with near-perfect accuracy, the vast numbers involved mean that inevitably, small errors, what scientists call mutations, will arise. Some might be as subtle as a single letter shift in those billions of bases, while others can involve entire stretches of DNA, added, deleted, or rearranged. These changes, though rare, are a driving force of uniqueness, adaptation, and sometimes vulnerability. Mutations come in many forms. The most common are point mutations, where a single nucleotide, the A, T, C, or G, is swapped for another, like a typo in a long book. Others are insertions or deletions, where an entire letter or even a sequence is added or lost. Rarely, large segments of a chromosome can be duplicated, deleted, or moved elsewhere, a phenomenon called chromosomal rearrangement. While the majority of these changes are harmless, and some are promptly repaired by your cell's built-in proofreading systems, a few leave lasting impressions, shaping evolution, creating new traits, and sometimes causing inherited disorders. A fascinating aspect of mutations is that they are natural and universal. They happen in all living things, from dandelions to whales, and are more likely in cells that divide rapidly, like those in developing embryos or regenerating tissues. Many mutations occur spontaneously simply because of the chemistry of DNA and the sheer frequency of copying. Others are encouraged by environmental factors. Certain chemicals, ultraviolet light, or even errors caused by radiation can overwhelm the cell's defense systems, inserting permanent changes into your genetic story. While some mutations have dramatic effects, such as causing sickle cell anemia, a single DNA-based substitution in the hemoglobin gene, cystic fibrosis, a deletion of just 3 bases, or Huntington's disease, a repeated genetic sequence, most changes are minor. Many are silent mutations, occurring in non-coding regions or making no difference in the construction of proteins. Some are even beneficial, giving carriers resistance against particular diseases or helping populations adapt to extreme climates, new foods, or emerging threats. For instance, the mutation that causes sickle cell can also confer resistance to malaria, a striking example of how genetic change and environmental pressure can work together in evolution. Sometimes the outcomes of mutations aren't seen in your lifetime but ripple through future generations, helping species, including humans, adapt and diversify. The very differences that make you distinct from every other person, your freckles, your exact height, your specific reaction to medications, can be traced to a patchwork of countless small mutations, quietly accumulating and blending over centuries. This interplay of change and stability helps explain why humans are at once so similar and so beautifully unique. Occasionally, mutations can be harmful and lead to inherited conditions or increase the risk of cancer when they disrupt the careful balance of cell growth and repair. Knowing about these risks, however, is leading to a new era in medicine, one where scientists can screen for, and sometimes correct, troublesome mutations, offering hope to millions living with genetic conditions. New technologies like CRISPR are opening the door to editing mutated genes, holding promise for the future, but also raising important questions about responsibility, safety, and ethics. The story of mutation is, ultimately, the story of life's restlessness, its drive to experiment, to branch, and to embrace both its triumphs and its challenges. With every tiny change, your DNA records not only potential risks, but also the possibility for resilience and discovery. And as you drift deeper towards sleep, remember, the gentle, ongoing dance of mutation has written not just the history of disease and difference, but also of adaptation, creativity, and survival itself. Now, let's ease our minds into the story of a modern scientific triumph, the Human Genome Project's big reveal, an international collaboration that forever changed how we understand ourselves at the deepest possible level. Picture a world only a few decades ago, where the full script of human DNA remained an enigma. Scientists knew there were genes, and even some that cause certain diseases, but the detailed sequence, the precise order of all 3 billion base pairs, was uncharted territory, like the darkened depths of an unmapped ocean. The Human Genome Project, often called the HGP, began officially in 1990, driven by the vision to read, letter by letter, the complete DNA of a human being. It was a grand scientific expedition involving thousands of researchers, dozens of countries, and a relentless drive to push the boundaries of technology and teamwork. Its stated goals weren't just to determine the sequence of the 3 billion base pairs, but also to identify all human genes, our genetic parts list, and to make this knowledge freely available to researchers everywhere. The challenge was monumental. Early DNA sequencing was painstakingly slow and expensive. Just determining the code of a single gene could take months. New machines had to be invented, and new forms of data storage were required to handle the flood of information. The project stretched across the United States, the United Kingdom, Japan, France, Germany, China, and more, uniting laboratories around the world. People like Francis Collins, who led the public effort, and Craig Venter, who led a competing private effort, became household names, celebrated for propelling us forward into the age of genomics. In 2003, after 13 years of quiet, persistent progress, the Human Genome Project delivered its first full draft. The achievement was breathtaking in its scale and importance. Never before had so much data about a single organism been gathered and analyzed. For the first time in history, humanity could scroll through its own recipe, letter by letter. We learned that humans, with fewer than 25,000 genes, were not so genetically abundant as once thought, but our complexity arose from how those genes were used, modified, and regulated. The data soon revealed an even more humbling fact. More than 99.9% of our DNA is identical between people, regardless of ethnicity, gender, or where their ancestors wandered. That tiny 0.1% is responsible for all visible diversity, proving that, at our core, humanity is deeply united by genetic kinship. The Human Genome Project was not just an academic victory. It opened the door to whole new branches of medicine. Researchers began to identify the exact genes involved in thousands of genetic diseases, setting the stage for diagnostic tests and, eventually, personalized treatments. Pharmaceutical companies began to design drugs for specific genetic profiles, and doctors could warn families of inherited risks or offer advice on how to minimize them. This ongoing wave of discovery is only accelerating. What once took years and billions of dollars can now be done in a day for just a few $100s, thanks to what this project unleashed. Sequencing your DNA is becoming as routine as a blood test, and the potential for early intervention, informed health planning, and new therapies grows year by year. Yet, even now, the Human Genome Project is just the beginning. Much of your genome remains mysterious, dark matter that does not code for proteins but may play important roles. Scientists continue to tease out which sequences turn genes on or off, how mutations influence your health, and why certain traits or illnesses run in families. The collaboration, data sharing, and curiosity that defined the Human Genome Project are now the foundation of what we call genomic medicine. So, as you rest tonight, consider the quiet revolution that began only a generation ago. Because of the Human Genome Project, nearly every question you could ask about your biology, ancestry, or risk for certain diseases can now be answered with breathtaking speed and precision. This modern epic of discovery has helped transform your DNA from a genetic script hidden in darkness to a story increasingly illuminated by human insight, collaboration, and hope. Let's shift gently now, letting science settle over you like a comforting blanket, as we consider the beautiful interplay between your genes and your environment, two collaborating authors responsible for your ever-evolving story. While DNA provides the blueprint, the unique sequences and instructions that govern your biology, it's the world around you and the life you live within it that weaves further details into the narrative. This partnership, known as gene-environment interaction, is a profound truth of how traits and destinies unfold in every human being. Your genes set the stage. They influence everything from the color of your hair and the shape of your face to how your body manages cholesterol or responds to infection. But these instructions do not act alone, nor are they rigidly set at birth. Starting from your earliest moments, indeed, long before you take your first breath, your developmental environment begins to shape which parts of your genetic script are read and when. Factors as simple as nutrition in the womb can alter how certain genes are activated, influencing birth weight, metabolism, and even long-term risk of certain diseases. As you grow, the environment continues to mold your biology. Exposure to sunlight triggers the production of vitamin D and affects how genes involved in skin pigmentation are expressed. If you exercise, you stimulate genes related to muscle growth, heart health, and even mental well-being, sometimes switching them on in ways that continue to benefit you throughout life. Diet, too, leaves its fingerprint. Certain foods can nudge genes involved in fat storage, energy use, and detoxification, resulting in outcomes as varied as food intolerances, tendencies to gain or lose weight, and even subtle changes in memory or mood. Stress, love, trauma, and learning. All of these shape the expression of your genetic code. Consider identical twins. Though they start life with nearly identical DNA, their unique experiences as they age can make them look, think, and feel remarkably different. This divergence is often visible not just in their personalities and preferences, but in the very molecules their bodies make, revealing the fingerprints of a lifetime's worth of environmental impressions. Scientists have uncovered thousands of examples where the environment makes a crucial difference. A well-known story is phenylketonuria, or PKU, a rare inherited disorder caused by a single faulty gene affecting how the body breaks down an amino acid called phenylalanine. Untreated, PKU leads to intellectual disability. However, if the condition is detected early and affected children avoid phenylalanine-rich foods, they develop normally. Here, the outcome depends on both gene and environment, the instructions and the context combining to shape health. This delicate dance does not stop at external exposures. Even the microbiome, trillions of bacteria and other organisms living in your gut, skin, and everywhere on your body, interacts with your human genes, affecting everything from inflammation to nutrient absorption, moods, and immune responses. The people you love, the air you breathe, even moments of sorrow or joy, all of these become signals, inviting your cells to adjust, adapt, and write new chapters in your genetic story. Understanding that genes and environment collaborate is profoundly hopeful. It means that heredity is powerful, but seldom absolute. While certain health risks may be written into your code, the choices you make, and sometimes those made for you by family and circumstance, can enhance, diminish, or sometimes even override genetic tendencies. So, as you settle tonight, breathe in the gentle assurance that your DNA does indeed script your foundations. But each day's experiences, choices, and dreams quietly revise your ongoing tale. You're not merely the sum of your parts, but the harmonious result of instructions and impressions, fixed letters and flowing life. Let's float even more softly, deeper into the subtle currents that tune and refine your story, the fascinating world of epigenetics. If you imagine your DNA as the complete library of instructions for building and running your body, epigenetics is like the staff of skilled librarians who decide which books are front and center and which are tucked out of sight. Epigenetics does not change the actual letters of your DNA. Instead, it adjusts how those instructions are read, using chemical tags and markers that act as dimmer switches, turning genes up or down, on or off, in response to the world around you. One of the most well-known mechanisms of epigenetic regulation is DNA methylation. Here, tiny molecules called methyl groups attach to specific areas on your DNA, usually at sites rich in a particular sequence, cytosine followed by guanine, known as CPG sites. When these methyl groups are present, the gene is typically silenced, its instructions harder for the cell to read. In other places, little chemical tags plane or modify the histone proteins around which DNA is wrapped. These changes make the DNA more or less accessible, allowing the gene's message to be copied efficiently or hiding it away. Think of these chemical tags as bookmarks or sticky notes, signaling which chapters of your genetic manual to focus on and which to skip for now. What's truly astonishing is that many epigenetic changes are influenced by the environment and can sometimes even be passed down to future generations. Diet, stress, exposure to toxins, levels of physical activity, and even periods of intense joy or adversity can leave epigenetic marks on the DNA in your cells. For instance, studies of children born to mothers who experienced famine during pregnancy have revealed lasting changes in the children's DNA methylation, affecting their own health decades later, including risk for diseases like diabetes and heart disease. Epigenetic changes play dramatic roles in health and development. In childhood, waves of epigenetic reprogramming switch on or off genes responsible for growth, metabolism, and the maturation of organs. During puberty, sweeping epigenetic shifts activate new genetic instructions, orchestrating development with exquisite timing. Throughout life, individual experiences, smoking, diet, trauma, exercise, continue to subtly modify epigenetic patterns, which is why identical twins with practically the same DNA often become quite different as epigenetic differences accumulate over time. And when epigenetic regulation falters, the effects can be profound. Many cancers are linked to abnormal methylation patterns that silence tumor suppressor genes, allowing unchecked growth. Other disorders, from Fragile X syndrome to certain forms of mental illness, appear when key genes are epigenetically silenced at the wrong moments. Even aging itself is thought to be guided in part by gradual changes in the epigenetic landscape, a slowing of some functions, and the chronic activation or suppression of others, like the fading of particular notes in a symphony over time. The exciting realization is that in some cases, these epigenetic changes are reversible. Certain epigenetic drugs are in development to try to reawaken silenced genes or quiet those that are too active. Studies in animals show that exercise, meditation, and particular dietary patterns may influence beneficial epigenetic marks. This brings a hopeful perspective. While your genetic code sets out the initial plan, your ongoing habits and environments continue to fine-tune how much of that plan is realized. Epigenetics bridges the worlds of nature and nurture in a way that transforms our understanding of health, identity, and even inheritance. Each night as you rest, your cells are adjusting, responding, and updating their instructions based not just on inherited DNA, but on the ever-changing epigenetic notes layered gently above it. So, as you close your eyes tonight, let yourself appreciate the flexible, responsive system quietly shaping your tomorrow, a blend of ancient script and living, breathing revision. Your story, written anew each day, is as much about what is possible as what is predetermined. Let's softly lift the curtain on a marvel of our modern era, DNA testing, which gives you, right from your home or doctor's office, a window into your hidden identity, ancestry, and even aspects of your health. Only a few decades ago, it seemed impossible that a single cheek swab or vial of saliva could unlock so many answers about who you are, where you come from, and what subtle traits you might carry. Today, Thanks to advances in science that built upon discoveries like the Human Genome Project, DNA testing is accessible to millions and has forever changed the way people view themselves and their unique stories. To understand how DNA testing works, imagine that vast genetic library inside your cells. Each testing company uses advanced sequencing machines or microarrays, impossibly tiny chips that can search for thousands, even hundreds of thousands, of specific letters or variants in your genome. In a forensics lab, these tests might focus on certain repetitive sections of DNA known as STRs, short tandem repeats, to create a genetic fingerprint unique to you, helping solve crimes or identify missing persons. In a medical context, tests look for changes in known genes that may signal increased risk for certain inherited diseases, such as breast cancer, mutations in BRCA1 or BRCA2, cystic fibrosis, or sickle cell anemia. Perhaps most popular and intriguing are the consumer ancestry and trait tests, with companies like 23andMe, Ancestry DNA, or MyHeritage now household names. These services compare your DNA with massive databases assembled from people all around the world. By analyzing specific markers scattered through your genome, they estimate the percentage of your ancestry coming from different regions, perhaps Northern Europe, West Africa, East Asia, or Indigenous America. The precision is never perfect and evolves as more reference data become available, but it's humbling and wondrous to see your genetic ties, sometimes stretching across continents and unearthing surprising connections. These tests can also reveal inherited traits you may not even know you carry. Can you smell a particular odor, like asparagus after eating it? Are you likely to have a unibrow or curly hair? Geneticists have located specific genes or combinations that strongly influence these characteristics, though of course environment and chance still play their parts. Some companies even offer wellness insights, like hints about your metabolism, lactose tolerance, or tendency toward deep or light sleep, all inferred from tiny differences in your DNA code. Medical genetic testing is especially powerful for families with hereditary diseases. Newborn screening, for example, seeks out dozens of rare but treatable disorders immediately after birth, giving children the best possible start in life. Diagnostic tests can confirm, explain, or rule out certain conditions in people with unexplained symptoms. For those with a family history of inherited cancers, heart disease, or rare genetic syndromes, knowing your risk can mean earlier screening, targeted therapies, or even life-saving lifestyle changes. Of course, DNA testing also brings important questions. Genetics can reveal long-held family secrets, undiscovered siblings, or unexpected histories that can be both joyful and challenging. Privacy remains a concern, as genetic data are particularly sensitive. Wise researchers, clinicians, and ethicists advise careful thought before pursuing answers and sharing results, reminding us that our story is both uniquely ours and intimately connected to the stories of our relatives. While it may seem almost magical, and sometimes a little intimidating, to uncover so much from a few cells, it's ultimately a tribute to the power encoded within your DNA. These tests pull back the veil on inheritance, ancestry, health, and even mysterious talents, offering clarity and sometimes new questions to explore. So, as you rest, remember that each cell in your body holds clues to mysteries both near and distant, waiting, should you ever wish, to be revealed by the gentle science of DNA testing, bringing your hidden story quietly to light. Drift with me as we enter some of DNA's hidden mysteries, the strange but true realities that underline just how surprising and sometimes counterintuitive genetic science can be. The more scientists uncover about genomes, the more we realize how much remains mysterious or unexpected. Within every cell's nucleus are puzzles, paradoxes, and phenomena that often leave even experts in quiet awe. For many years, biologists believed that every piece of DNA in our genomes had a purpose for coding proteins, the key machinery and structures of life. Yet, as the Human Genome Project revealed, only about 1–2% of the human genome actually contains these protein coding instructions. So what about the other 98–99%? Early researchers were perplexed, labeling this vast expanse as junk DNA, thinking it to be leftover detritus of evolution. mere filler in the recipe book. But as research deepened, scientists found that large portions of so-called junk DNA serve subtle but critical roles. Some segments help control when and where genes are turned on or off. Others act as switches, enhancers, insulators, or templates for different types of regulatory RNA molecules. Some stretches are repetitive, echoing sequences that may influence chromosome structure or stability, and others seem to be ancient viral DNA, echoes of infections suffered by distant ancestors now woven permanently into our chromosomes. Even more astonishing, you share a surprising amount of DNA with seemingly unrelated organisms. Consider the banana. Humans and bananas share about 60% of their DNA. This doesn't mean you are mostly banana, but that many genes responsible for fundamental cellular processes are common to both of you and the humble fruit. With mice, the overlap climbs to nearly 85%. Between different people, genetic similarity is even greater. Any two unrelated humans are about 99.9% genetically identical. Just 0.1% carries all the individual differences in height, appearance, disease risk, and personality. Another marvel is the phenomenon of genetic jumping genes, or transposable elements, first discovered by Nobel laureate Barbara McClintock in maize, corn, in the 1940s and 50s. These are DNA segments that can change their position within the genome, sometimes creating or reversing mutations and altering genetic identity from one generation to the next. Nearly half of all human DNA may be made up of these restless, mobile elements, suggesting your genetic story is written and sometimes rewritten by moving passages within your own cells. There are other strange phenomena too. A single gene can sometimes create multiple proteins, depending on how its instructions are edited, a process known as alternative splicing, vastly increasing the proteomic diversity possible from a relatively small gene count. Then there's the wonder of mitochondrial DNA, a tiny circular genome contained in the power plants of your cells, inherited almost solely from your mother, tracing maternal ancestry far back in time. And perhaps most humbling of all is the fact that, despite the enormous database of knowledge we now have, more than half of all human genes have functions that remain mysterious, marked simply as predicted protein or unknown, waiting for future discovery. The story told by your DNA is not finished. It is a living text, forever open to footnotes and revelations. As you drift further into restful thought, remember, your DNA contains universes, from ancient viral echoes to sequences with no known purpose, from similarities with all life to the one-of-a-kind patterns that set you apart. Each mystery whispered by your genome is an invitation to wonder, and a gentle reminder of how much magic remains even in the most precise science. Let's glide now into a realm where DNA knowledge becomes medicine, where learning your genetic code leads not just to understanding, but to hope and healing. The science of genetics has transformed medicine from a one-size-fits-all approach to a more tailored personal journey, often called precision medicine. In this gentle revolution, your DNA serves as a map, offering clues, warnings, and helpful guides so healthcare can work with, and not against, your unique biology. Consider the traditional model of prescribing medication. For generations, doctors offered the same drugs at the same doses to every patient with a similar diagnosis, based on large studies, averages, and best guesses. Yet we now know that particular gene variants can dramatically affect how your body processes drugs. For example, a gene called CYP2C19 helps the liver break down certain medications, like some heart drugs and antidepressants. If you carry a version of this gene that works slowly, you may need a much lower dose or even a different medication to get the right effect and avoid harmful side effects. This knowledge is now saving lives, thanks to a new field called pharmacogenomics. For some diseases, knowledge of genetics has directly changed the outcome. Take cystic fibrosis, a genetic illness affecting the lungs and digestive system. By understanding the precise mutation in each patient's CFTR gene, doctors can prescribe targeted therapies that correct the faulty protein, sometimes transforming lives once dominated by chronic illness. The same is true for certain cancers. Breast, ovarian, and colon cancers often run in families because of specific inherited mutations. With DNA testing, at-risk individuals can receive more frequent screenings or preventive care, sometimes even taking medications or having surgery before cancer ever appears. Genomics also helps solve medical mysteries that, long ago, were matters of despair. For children born with unexplained developmental issues or rare diseases, rapid whole genome sequencing can sometimes point to a diagnosis and, in rare cases, suggest a meaningful treatment. Newborn screening, quietly performed in hospitals around the world, now identifies conditions like phenylketonuria, PKU, before symptoms appear, allowing dietary changes or therapies that protect the child's health for life. Vaccines and treatments for infectious diseases are also touched by genetics. Analysis of the COVID-19 virus's genetic code, for example, allowed scientists to develop vaccines in record time and track new variants as they arose. Some individuals have genetic traits that make them more or less vulnerable to specific infections or side effects. Researchers are exploring treatments for HIV, sickle cell anemia, and even inherited blindness that harness knowledge of the DNA code, rewriting or editing it using tools like CRISPR. Looking to the future, the dream of personalized medicine glows ever brighter. Imagine treatments for cancer targeted precisely at the tumor's unique mutations, or rare diseases corrected before a child is ever born. Imagine drugs chosen because your specific body will use them efficiently and safely. Or gene therapies that can replace, repair, or silence broken genes before illness arises. Slowly, quietly, these possibilities are becoming realities, with clinical trials testing cures for conditions once thought impossible to change. Yet in all this, a gentle reminder. Your DNA may predict tendencies or risks, but very often it is only part of your story. Lifestyle, environment, chance, and kindness all have their roles to play. Still, the comfort offered by genetic medicine is profound, giving hope, ending some forms of uncertainty, and linking you through science to a future where care is as unique as your own cells. So as you relax tonight, let yourself take peace in knowing. Within the code quietly written in your cells, the future of healing is being quietly prepared, more gentle, more effective, and always more personal. As you become even more restful, let's gently marvel at a delightful paradox of your genetic story. Every cell in your body, every skin cell, muscle cell, neuron, or blood cell, carries essentially the same DNA, the very same complete set of instructions that first sparked the development of your unique human form. And yet, when you look at yourself in the mirror or think about the wondrous complexity within, your eyes that see, your heart that beats, your bones that move, each of these structures is strikingly different, exquisitely specialized for its own role. How then can a single unchanging code give rise to such spectacular cellular variety? The answer lies in the artful choreography of gene expression. Imagine each cell as a musician given the same symphony sheet, but led to play only the notes and at the volumes suited for that part in the orchestra. In other words, even though every cell holds your entire DNA script, only a fraction of the genes are switched on in each cell at any given time, while the rest remains silent. For example, cells in your liver activate genes for detoxification and metabolism, but barely touch the instructions for making hemoglobin, which are switched on in your red blood cells. Skin cells read the code for keratin and melanin. Neurons focus on instructions for building neurotransmitter machinery. This exquisite system starts taking shape early, as your first cells divide after conception. Chemical signals, spatial cues, and a cascade of interacting molecules guide the fate of each cell. As the embryo grows, a process called cellular differentiation occurs. Waves of signals nudge some cells to become part of the heart, others to form the brain, or to branch out into skin, hair, or the lining of the gut. The DNA itself is unchanged. The instructions are all still there, but the pattern of gene expression becomes unique to each cell lineage. Epigenetics, which we considered earlier, is a crucial part of this process. The addition and removal of chemical marks guide cells in deciding which genes to read and which to ignore, creating a lasting memory that is passed on to all future cells of that type. This is why a single fertilized egg can eventually produce the dozens of cell types that make up your body, each variation tailored by a layer of decisions about what parts of the shared genetic story are revealed. On rare occasions, this versatility can lead to errors. If a control switch malfunctions, a cell might read the wrong instructions, sometimes leading to diseases like cancer, where growth genes are turned up too loud or stop signals are ignored. Yet most of the time, the system works quietly and beautifully, orchestrating the turnover and renewal of your body for a lifetime. There's a quiet majesty in reflecting on this. When you notice the diversity in nature, from the petals of a flower to the neuron in a hummingbird's brain, much of it comes down to this same principle. Life does not need endless different blueprints for endless different forms. It needs a single script, interpreted with boundless creativity. In you, the story plays out in parallel, every moment across trillions of cells, each cell a participant in the grand performance that is your living body. As you savor the final moments before sleep, let your thoughts rest in the calm assurance that, though each cell is reading the same DNA story, the outcome is as rich and multifaceted as life itself. Every heartbeat, every thought, every breath is proof that unity and diversity can arise together from a single silent code. Sink comfortably into the future of your genetic story, where the boundaries of possibility continue to expand, shaped by the whispered promise of new discoveries and technologies. For most of human history, our genetic heritage was a silent script, unread and unchangeable. But now, dizzying advances are allowing us not only to read, but even to consider editing the foundation of our biological selves. One of the most hypnotic developments of recent years is CRISPR, an elegant gene editing tool discovered in bacteria, harnessed by researchers Jennifer Doudna and Emmanuelle Charpentier, who were awarded the Nobel Prize in Chemistry in 2020. CRISPR acts like a pair of molecular scissors, able to seek out a specific region of DNA and cut it with incredible precision. This opens the possibility of removing, replacing, or even adding genetic sequences, much like editing a word in a novel. While earlier methods of genetic modification were slow, imprecise, and unreliable, CRISPR makes gene editing faster, easier, and potentially much safer. Today, researchers around the world use gene editing techniques in laboratories to study diseases, test new drugs, and even attempt to cure inherited disorders in animals. In some rare instances, early clinical trials in humans have shown that editing a single faulty gene can eliminate symptoms of diseases like sickle cell anemia or certain types of genetic blindness. Each successful trial, each gentle fix, is a glimpse into a future where medicine may be able to repair our code, not just treat its consequences. But as with any powerful tool, CRISPR, and the gene editing revolution it signals, raises questions as deep and wide as the science itself. When should we intervene? Should changes be limited to treating disease, or could they extend to enhancements like stronger bones or sharper memory? Can we ever fully predict the consequences of altering genes that have evolved over millions of years? Ethical voices urge careful, slow progress, reminding us that even tiny edits may echo through generations. Beyond editing, the future of DNA holds other marvels. Scientists are exploring gene drives to control invasive species, synthetic biology to craft organisms that clean pollution, and even resurrection biology to study extinct creatures by reconstructing their genetic code. Some visionaries whisper about whole genome design, where prospective parents could select or edit genetic traits, a scenario that is both hopeful and deeply cautionary. At the same time, your role as an individual genetic storyteller will only grow. With ever cheaper sequencing, you may soon know your genome as well as you know your blood type. Medical care may become ever more personalized, responsive not only to your needs, but to those encoded possibilities within your DNA. Gene therapy, targeted drug regimens, and risk assessments woven into wellness plans may become the quiet norm, keeping you healthy for longer than was once imaginable. Yet, for all the excitement and possibility, the greatest comfort may lie in knowing that your DNA's story is more than the sum of base pairs. Each evening, as you prepare for rest, remember that these advances build upon an ancient script, humming quietly within every cell, a story both timeless and, now, responsive to gentle human hands. So, as you dream tonight, let yourself float on the gentle current of this future, a future shaped by care, caution, and curiosity, where the story of you, written in DNA, is slowly becoming a story you help to write. Let your thoughts settle now, gently winding down into the most intimate and enduring truth of all. Your story is written, cherished, and maintained in every cell, even as you drift into sleep. Each night, as you grow still and the world quiets around you, your DNA continues its silent work, repairing, copying, and orchestrating the living symphony that sustains you. The cells in your body never truly sleep. While you lie restful beneath your blanket, they quietly divide, read, and rewrite tiny segments of your genetic script, keeping you alive, healing you, and preparing you for the day to come. Consider the nightly tasks of DNA repair. Small teams of molecular caretakers patrol each strand, searching for damage caused by daytime stress, sunlight, or the simple wear and tear of being alive. With soothing patience, these repair enzymes snip out mispaired bases, stitch up broken stretches, and gently smooth over frayed ends, ensuring that the precious story written within you remains as accurate and complete as possible. These repairs are vital. Without them, mistakes could pile up, jeopardizing the health and stability of each cell. The process is so seamless, so exquisitely coordinated, that you rarely have any sense of all the careful editing still happening while you sleep. As you move through each night's cycles of deep rest and dreaming, your DNA is at work in less obvious but equally essential ways. Hormones released during sleep trigger waves of cellular rebuilding, the activation or silencing of genes needed for growth, learning, or emotional healing. In children and adolescents, deep sleep is when growth hormone encourages the unfolding of genetic blueprints for muscles and bones. For adults, restorative sleep strengthens immune function, guided by genes that tell immune cells when to renew and prepare for tomorrow's challenges. Even your dreams, shaped by new memories and quiet reflection, may weave around genes involved in memory and plasticity. These genetic signals help strengthen the neural connections you need to learn, adapt, and make sense of the world. Your DNA might not write your dreams, but it ensures your brain cells have exactly what they need to make dreaming and learning possible. The peaceful work of your DNA at night is, in a sense, love in molecular form. Tireless, silent, always protecting, repairing, and renewing the essence of your unique self. It's a story without a final page. Each sunrise begins a new chapter, shaped by your experiences, choices, and the wondrous collaboration of code and life itself. So as you lie in the softness of night, know that your DNA keeps a gentle vigil, caring for you with every breath and heartbeat. Your genetic story, a story of ancestors, mysteries, hopes, and possibilities, never rests, always working to keep you whole, healthy, and ready for another day. And now, as we come to the end of our gentle journey, I want to thank you for spending this quiet time learning and resting with me. I'd still love to know where in the world you're watching from and what time it is there. I find it wonderful to imagine people wrapped in comfort and curiosity all across the globe, joined together by the simple wonder of science. May your sleep tonight be deep and healing. May your cells find peace in their quiet duties. And may the story written in your DNA, patient, caring, and uniquely yours, guide you softly into tomorrow. Sleep now, and let your dreams drift through double helixes and starlit code as the whispering science whispers your story into the night.